So, any microbe that is related to some sort of infection or results in an infectious disease is called a pathogen. And the pathogenicity of that organism is the definition of like what the severity of the infection is that they can cause. So something that has a high pathogenicity is really going to cause a more severe infection. Now, there are some organisms that are considered true pathogens and some that are considered opportunistic pathogens. So I sort of already touched on the idea of opportunistic pathogens in the last video, but I didn't really talk about it in those terms. So we talked about an endogenous infection. So an endogenous infection was one where you had two organisms that were sort of growing together within the body. And when one of those organisms became depleted or was compromised, then the other could opportunistically begin to grow and then cause an infection. So that would be the case of an opportunistic pathogen, one that will become a pathogen or if the opportunity arises. Whereas a true pathogen is one that will always be an infectious causing agent. All right, so really whether or not you get sick from exposure to a microbe has a lot of different factors about whether or not it's really going to work to cause a disease, right? So there's some microbial factors and then there's some host factors. And I'm going to walk through all of these, but this sort of shows all of the different types of things that we have to think about. So in terms of the microbe, we have the virulence. So the virulence, and again, I'll talk about this a little bit more in upcoming slides, but virulence is really sort of how dangerous you are. What kind of factors, what kind of things do you have that make you a good pathogen? The percentage of the optimal dose. So an optimal dose is sort of the exposure rate. Do you have to be exposed to a high concentration of this pathogen or a low concentration of this pathogen? There's some optimal amount of bacterial exposure that you need to have for when an infection really will occur. And then the correct portal of entry. Does that bacteria get in through your lungs or does it get into a cut in your skin? Right? So is it something that you eat, right? So some things will only be pathogenic or disease causing if they enter into your body in the right way. Then we have the host factors. So you as a host have lots of different ways in which you can fight off an infection or prevent a disease. And so the host factors that are contribute to whether or not you get sick is your genetic profile. So whether or not you are susceptible even genetically to that. And then the function of your non-specific host defenses, which we'll talk again more when we get into immunology. And then whether or not you've been exposed to that microbe before. If you have been exposed to that microbe before, you have some memory of that and your body might be good at fighting that off. And then lastly, just sort of your general health. Okay. So with all of these things sort of coming in and aligning, there's three possible outcomes. The microbe could go through your body and pass without notice. So you might not have any sort of infection or establishment of infection. And this happens quite often. Your immune system is pretty good at fighting off anything weird that comes in that's not you. Sometimes the microbe can establish itself and it doesn't cause disease. And then, the worst being that the microbe would eventually cause the disease. So virulence is really the degree of the pathogenicity. It's how dangerous that microorganism is. And this virulence is determined by how well that bacteria can get in and cause damage within the body. And all sorts of different types of things can be considered virulence factors. So it's really sort of anything that allows it to achieve one of those two things, establishing itself within the host and then causing damage. So sometimes it's a structural component. So like fimbriae can be considered a virulence factor because it's like these hair-like bristle structures that allow you to adhere to the infection site. Or the production of toxins can be a virulence factor where those toxins would cause damage to the tissue. All right, so there's really there are three steps to an organism becoming established within the body, and then we'll talk in an upcoming video about actually then it causing infection. So the first step is the portal of entry. So the organism has to get into the body, and it really has to get into the body in the correct portal. So most often this is going to happen between on the skin or some sort of cut in the skin, or through a membranous boundary. So that means like your nasal cavity or within your lungs, somehow in your mouth, your eyes, your ears, your urogenital tract. So these are places where you might be exposed to a bacteria from the outside world, and then it would be able to establish itself, right? So any of these sorts of infection are called exogenous, as opposed to those endogenous ones that we were talking about were already within the body. Right, so you can enter in through the skin in terms of like nicks or cuts or abrasions. The gastrointestinal tract, we can get a pathogen into the body by consuming food or drinking contaminated water. And these organisms specifically will have to be adapted to withstand low pHs, right? So, because your stomach is full of stomach acid. So immediately most organisms that you consume are going to die upon entering into your stomach. But some pathogens can withstand the pH of your stomach. Your respiratory tract, this is the greatest number of pathogens. So anything that's in the air that you can breathe in. And then through your urogenital tract via sexually transmitted diseases. Now I mentioned this a little bit last time. There are some organisms that can actually pass into the placental or pass across the placental barrier, but this is pretty rare. Most organisms are prevented from moving into the womb, but some can move into and get a baby or a fetus can be exposed to certain types of microorganisms. Toxoplasmosis, like that's the one where you can be exposed to it if you're near like cat feces. Ugh, cats. Anyway, but this is sort of an acronym to sort of keep in mind some of the organisms that can cross that placental barrier or can contaminate children during birth. And we also, when you look at the infectious disease lectures you're supposed to cover on your own time during this class, it goes into different diseases that babies can acquire as they move through the birth canal. Right. Now, the next is the size of the inoculum. So really, how much of the bacteria are you exposed to? And for every different type of bacteria, in order for it to cause an infection, we have something called the infectious dose. Only if you are exposed to a certain number of bacteria are you going to establish infection. And for things that have a smaller infectious dose, they're considered to be more virulent because it takes less of them to do damage or to establish the infection. All right, so that's portal of entry, sort of like gaining access. Now, once the bacteria actually get into the body, they also then have to stay, right? So they have to attach to the host somehow through the process of adhesion. So we have looked at some different adhesion factors when we've looked at the structure of prokaryotes and the structure of viruses. So fimbriae are those little hair-like structures that might help you to cling onto a host cell, or maybe the lining of the intestinal wall, as well as capsules and spikes on viruses. So those are all things that help you to establish your presence in the host. And then the last thing you have to do is you have to be able to survive there. So in order to survive, you have to avoid host cell defenses. And most commonly, you're going to have to avoid phagocytic cells. So phagocytic cells within a host their job is to really identify foreign things and to engulf them into the cell and then to attack and kill them within the immune cell. So lots of different kinds of white blood cells, macrophages, neutrophils, and again, we'll get into this more in detail when we get into immunology, are sort of circulating around the body and trying to identify and destroy anything weird that comes in. So some things that bacteria do to avoid this is they can produce things called anti-phagocytic factors that prevent them from being phagocytized by host cells. So leukocidins are chemicals that are made by strep and staph that are toxic to white blood cells. They might have an extracellular surface layer so they can kind of hide themselves and can't be found by immune cells. But also some of them are good at surviving inside the phagocytic cells. So mycobacteriums, tuberculosis specifically, can be engulfed within immune cell, but then still survive once it is in the immune cell. So those are really the 3 steps at first to getting into the body. So you have to have the correct portal of entry and the correct infectious dose. You've got to be able to stick within the immune cell, and then you have to avoid host defenses. In the next lecture, I'll actually talk about the properties that you have or need to have in order to then move forward and begin to cause an infection.